Okay, so we are going over my notes for life science and cell biology in a nutshell. We're learning about how cells divide, and we already discussed the the role of how the the fibers inside the cell, also known as um, the spindle fibers and the aster, everything inside the cell uh, moves around and is pulled apart during cell division. So we discussed the steps in mitosis. So and now we're going to go over meiosis, which is similar to mitosis. It's also cell division. It's only different is the type of cells that undergo meiosis. And so you go through the phases of G1, which is increasing the amount of mass of the cell that is going to be duplicated. This is in mitosis. The S phase synthesizes DNA, making it more. G2, more proteins and more cell mass is created. Then through prophase, you have the chromosomes coiling up. The sister chromatids of each chromosome joining at a centromere. The mitotic spindle and the asters around the spindle and pulling apart the centriole pairs. Um, so um, all the chromosomes are pulled apart, and finally in late prophase the nuclear envelope is broken up, and metaphase you have the lining up of the chromosome molecules. Anaphase, you have separation and the movement to opposite ends of all the chromosomes. And in telophase, you have a new nuclei forming around the two groups of chromosomes on opposite ends of the cell. Um, so, so let's see. So that's how the cytoskeleton helps with cell division. All these fibers and protein fibers. So in contrast, in meiosis you have production of gametes. So again, you're going through the same stages essentially, except telophase is gone through twice in order to produce 4 haploid cells at the end instead of just two. So instead of 2 diploid cells like in mitosis. So in sexual reproduction, so involving sexual cells rather than body or somatic cells, haploid gametes unite. When you have fertilization, you need 2 haploid gametes becoming a diploid zygote, which is basically like the baby. So you have to have haploid gametes, otherwise you would end up with the wrong number of chromosomes. So you see in the parents you have to have a haploid number of chromosomes, so half the amount, and the 2 parent cells joining together to form the zygote or the offspring cell is going to be a normally numbered diploid number, same number as that the parents had in their cells. The zygote continues to divide by mitosis and develops into a fully functioning new individual. So basically, meiosis is for the purpose of recombination of genetic material of the two parent cells into a zygote cell, or the offspring cell. The initial cell is called a zygote, and once you have that, it divides to develop into a fully formed organism by the process of mitosis, which we discussed in previously. So, and meiosis is just simply just one cell dividing into two cells, or increasing the number of cells basically by duplication. And the summary is the spindle apparatus consisting of microtubules and proteins manipulate chromosomes during cell division. The spindle fibers attach to replicated chromosomes, moving and separating them when appropriate. The mitotic and meiotic spindles move chromosomes to opposite cell poles, ensuring that each daughter cell gets the correct number of chromosomes. The spindle also develops the location of the Mitosis plate. The centrally localized site becomes the plane on which the cell eventually divides. In animal cells, the spindle apparatus determines the location of an important structure called the contractile ring. When it divides, it is formed from actin microtubule filaments and proteins, including motor protein myosin, contracting the ring of actin filaments, forming a deep groove called a cleavage furrow. So the myosin helps with that. As the contractile ring continues to contract, dividing the cytoplasm, pinching the cell into two along the cleavage furrow. The plant cells do not contain asters or star-shaped spindle apparatus microtubules, which help determine the cleavage furrow that animal cells have. No cleavage furrow is formed in plant cytokinesis. Instead, daughter cells are separated by a cell plate formed by vesicles that are released from Golgi apparatus organelles. The cell plate expands laterally, fuses with the plant cell wall, forming a partition between the two newly divided daughter cells. As the cell plate matures, it eventually develops into a cell wall. Daughter chromosomes result from separation of sister chromatids, as in all cells, occurring in anaphase of mitosis and anaphase 2 of meiosis. Daughter chromosomes develop from the replication of single-stranded chromosomes during the synthesis or S phase of the cell cycle. And as in all cells, following DNA replication, the single-stranded chromosomes become double-stranded chromosomes held together at a region called the centromere. Double-stranded chromosomes are known as sister chromatids, which eventually are separated during the division process and equally distributed among newly formed daughter cells. Each separated chromatid is known as a daughter chromosome. Here you have the showing of how these genetic material are separated using the spindle apparatus, which is in green, and the genetic material in the bright blue form, which are pulled apart to opposite ends. You have separation of one cell into two, and all the contents separate once the cleavage furrow splits up the cell into in the cytoplasm. All the organelles, everything is split into two, and this is shown from the external viewpoint. This is the membrane, how it looks like, and all the fibers surrounding. Mitosis versus meiosis. These two cell division processes are similar but distinct. Both processes involve division of a diploid cell or a cell containing two sets of chromosomes, one chromosome donated from each parent. Thank you.